Okay, you can go.
Okay. Well, hello everyone and welcome. Thank you for being here for today, for this introductory webinar. We are excited to give you a sneak peek of what's coming up in our upcoming webinar, upcoming workshop, sorry. Just a quick reminder about the aim of the IUVSTA events. Can you see the slide? Yes.
No.
No,
No?
no.
Not yet.
Okay.
Okay.
Yeah.
Okay, thank you. This is the workshop, characterization of gas transport through materials, application to vacuum science and technology, that will be held in October in Portofino, Genova, Italy. The workshop aim is stimulate discussion and collaboration between different scientific areas where gas transport through materials is a common topic, and vacuum science and technology make a significant contributions, especially focused on application related to the more relevant global environmental issue and sustainability. These are the main topics. Gas transport model, a part of theoretical part with dense and porous materials, phenomenon of absorption and desorption at surfaces, and then application in vacuum science and technology like insulation, component characterization, and outgassing, as well in the membrane and thin film technology, for example, hydrogen storage, CO2 capture, gas separation, food packaging, and so on. This is to have a quick reminder. During this webinar session, I'll have some questions for our invited guest. And at the end, there will be an open question and answer where you can ask anything would you like. Feel free to jot down your questions as we go along and using chat to set them to us. First, a big thanks to our invited guests for their availability. You will have about from 5 to 10 minutes to answer, focusing on the topics addressed during the workshop. No need to be exhaustive, just enough to spark interest among participants. You can share your slide if you want, or I will do it myself, as you prefer. There's no technical issue about this one. You can share slide by yourself. Okay. Let's start. Joining us now is Elsa Battista. Elsa Battista is the head of the Volume and Flow Laboratory of the Portuguese Institute for Quality. And this is my questions. Elsa, you have been responsible for the Volume and Flow Laboratory at the Portuguese Institute for Quality since 2002. Recently, your field of interest has focused on metrology standard in microfluidic devices.
No.
Do you have experience dealing with issues related to gas or liquid permeation leaks through polymeric microfluidic devices?
Thank you, Giuseppe, for your question. So I'll start sharing my screen so I can answer your question. So I'll do that now. And then I hope you can see the slides, and I'll put it on the vision mode. Yeah. So and then I start with this one. Yeah, so your question is interesting because I can answer in two parts. So first, you are correct. So I am the coordinator of a project which is entitled Establishing Metrology Standards in Microfluidic Devices. And this is the second time we got funds for this project. So we had a project that lasted 3 years, ended in May last year. And now we got funds again by EUROMET, the European Association of Metrology Institutes, to continue the investigation in this work. And in the previous project, as you can see here from this presentation, from this slide, we developed a guideline for testing leakage in and burst pressure in microfluidic devices. And we did that by establishing several protocols. And we have these interesting flow charts where we have a series of questions, and we decide on the different protocols according to the answer to the question. And in this specific project, we focus on protocol B, which is actually to measure leakage in a capillary flow device, so microfluidic device. So as you were saying, I have some experience in leakage of liquid in microfluidic devices and made of polymer, which is topaz, and also made of glass and PDMS. So from this protocol, we actually measure some leakage in a specific device, microfluidic device. So the sample was made according to here this picture, using the front track system, and we could see using pressure controllers and flow sensors the leakage we had from a specific channel. So we imposed a specific pressure, and we could see if it suit the pressure or not. This work was actually one of the deliverables of the project. And there is here a link that you get to all the information that we had. So I have some theoretical experience on leakage, mainly in liquids, but in gas, I actually don't have any. So, and it was interesting that you gave me this challenge because when you give me the challenge, I could see that it really fitted within this new project, which is the second part of the MFMET project, and it was just started. We have some, I think Federica, we need to, can you muted her, Giuseppe?
Yeah. Okay.
Thank you. That's better.
Okay.
So for this project that just started in June, which I'm also the coordinator, we have now one work package, which is general guidelines for quality control, validation, and characterization of microfluidic devices. And in this one, we actually have now one specific issue, which is new detection methods using liquids versus gases, and develop a guideline for measurements also at high temperature. So this is where it fits within your question and this conference, because we are now starting to study how can we actually measure leakage of liquids, but using gas, and then make all the theoretical calculations to convert the leakage from gas to liquid. So we will have just a special work package that is really related to this situation. It's something new. We have never tried it, so it's completely new. We just started the work. So in my presentation in October, I think I already would have some more information to give you, because we will start the investigations now, and within 6 months, I think we will have some more thing to show you. But at least we'll have I can show you the part of the liquid leakage. That is something we have done. And for the gas is really new, but we are very excited, and we have a lot of experts from PTB from Germany, and also in RIM from Italy, which are really used to work with gas. It's very small. So we are talking about leakage, which is like 1 microliter per hour or less. So very small thing, very small. And they are used to measure this kind of flow rate for very small quantities. And so we are now going even smaller. So it's a challenge also for us, for this community of national metrology institutes, universities, and also some manufacturers of chips to try and now use the the technology for gas and apply it to liquids in leakage,
Yeah.
and let's see if it works. So it's really a challenge and something a risk we are taking. But I think maybe even during the workshop or the conference, we can have some ideas if in case we are
Yeah.
not following the right path. So
Sure,
my presentation
sure.
will focus on these two things: first, the part of the liquid leakage, and then also the part of the challenge for liquid versus gas.
Okay. Thank you. Have you worked with Carl Houston of PTB, no?
No, not yet. No,
Ah, no? Okay.
no.
Because I've cited PTB in Berlin, the physical
Maybe,
technician.
yeah, maybe he's in the group, but it is Mateos who
Okay.
I'm working with from BTB, so it's
Okay.
a different person. But maybe they are
Okay.
in the same, because
Yes.
it is pressure and vacuum, the group that is also taking part of this
Yeah.
project now
Okay.
from BTB.
Okay.
So I hope this more or less answers your question. So yes, I have some experience in
Yes.
half the thing you asked, the other half I don't. So only theoretical things I have read and investigated.
Thank you, Elsa. It's very interesting, and I'm looking forward to see you in Portofino in next October.
Okay,
Yeah, very
thank you.
interesting. Then, if Hajime Yoshida is not still with us, I I can introduce Paolo Chigiato. Paolo Chigiato is a vacuum and material engineer in CERN, Switzerland, Geneva, where he holds the role of Deputy Head of Technology Department, Group Leader, and Program Coordinator of the Einstein Telescope, with recognizing international experience in vacuum technology, thin films, and surface finishing and thermal treatments. This is the question, Paolo, regarding hydrogen outgas. Hydrogen outgassing rate for stainless steel remains in the range 10 to minus 13 millibars liters per second per square centimeters after a backout at 300 degrees. To reach 10 to minus 15, vacuum firing typical required. However, which technique do you use to measure such extremely low outgassing rate? Wow.
Thank you, Giuseppe. I hope you can hear me.
Yes,
Okay,
yes.
thank you.
Hi.
So I have a small presentation here, a short presentation
Yeah.
for you. Let me know if I have to swap. I think yes.
Okay. Okay.
Okay, so think of the problem of measuring low rate of gas release
Yeah.
is not only a matter of hydrogen of stainless steel or fire stainless steel. It is also a problem that is quite general in vacuum technology. So here you see an example. Maybe I have to use the
Yes.
pointer. Where is it?
Yes, we see.
Wait a
At the
second.
bottom. Yeah. Okay.
Okay, doesn't matter. So here you can see with
Yes.
the typical outgassing rate
Yeah.
of measure here in torr-liter second square centimeter of a slab of different
Yeah.
variable thickness. Okay, for hydrogen after firing at 950 degrees for 2 hours in a furnace
Okay.
with a ultimate hydrogen pressure 10 to the minus 5 torr or millibar, let's say,
Yes.
at that temperature. What you see is that when the slab is thin, lower than
Yeah.
5 millimeters, sorry.
Yeah.
Okay. What you see is that you have outgassing rate, in fact, in the 10 to minus 15 millibar liter
Yeah.
second. Another typical case that is quite interesting in accelerators or systems that are massively pumped by non-evaporable getter
Yes.
is the release of noble gases, because noble gases are not pumped by the getter.
Yes.
So we are interested on that. And here you see that we can measure as a function of temperature the release of krypton from a film of titanium, zirconium, vanadium.
Yeah.
And you see that we can measure something of the order of log 10 to the minus 19. So we can measure with a quite elaborated process, something like 5 molecule of krypton per second per square centimeter. This is feasible. Another application is extremely low leak rate. For example, for gyroscope that are used in aeronautic or spatial industry, they require the measurement of helium leak rate lower than 10 to the minus 14 millibar liter per second. Okay, so it's always interesting to measure very low quantities.
Yes.
So now, well, the point is that the technique that are in general used for the measurement of the gassing rate do not work for such value of a gassing rate. Here you have the very standard is the accumulation method, where you monitor the pressure continuously during the accumulation of the gas. In general, you have a linear increase for gases that are not absorbed by the wall of the system. Okay, and the system has some intrinsic limitation. For example, you cannot measure the composition of the gas, so you collect everything. Okay, and the sensitivity here of the system is limited by the outgassing rate of the vessel and also by the gauge. And as I told you before, if you have a species like water vapor, which is very interesting to measure in terms of gassing, you cannot measure because there is a continuous pumping. So you attain an equilibrium pressure in between what is released and what is absorbed. The other method is the throughput method. Here you have your sample or a chamber attached to this flange here, and you measure the delta P toward a conductance of a known conductance. So the degassing rate is the difference of the pressure multiplied by the conductance. You divide it by the surface area. So in this system, the sensitivity is strongly limited by the fact that you have gauges that degasses a lot. Even the best gauge degas as a square meter of austenitic stainless steel, which is fired. Okay. And also the vessel around the sample can have a contribution which is important. Now, since 1995, here at CERN, we use a method which is a combination of the two, and in some sort, we can avoid most of the problem I mentioned to you before. In fact, what we have is a vessel that can be also the sample, if you needed to measure the vessel, which is extremely degassed as much as we can, vacuum fired, air fired, whatever it is. And this sample is connected to a measurement system by a valve which is itself vacuum fired to attain a very, very low outgassing rate. This is a variable leak valve, so you can open the valve very slowly. So what we do in general, we put the sample there as much as we can, okay, if the degassing rate is very low, or we use simply the sample as the chamber for which we have to measure the degassing rate. We bake, we pump continuously, okay, and then at a certain time, we close this valve and we let the gas accumulate. So the only gas which is accumulated here is the gas coming from the sample, the gas coming from the vessel, and the gas coming from the back part of the valve. Okay? So for those knowing the Pfeiffer or the variable leak valve is the sapphire of the valve. Okay, so Then at a certain point, certain number of hours or minutes, depending on the gassing rate, we open this gently, this valve, and we have a peak of gas, okay, developing in the measurement chamber, okay, until it attain a new equilibrium between the system. So this gas expanding in the measurement system is detected by residual gas analyzer, which is calibrated, and a Balmer gauge, which is low outgassing rate, but okay, is not really important because it's not inside the sample accumulation system. And this part here, to attain a good sensitivity, is kept to a very low pressure, 10 to minus 11, 10 to minus 12, measured by this, pumped by these through these conductance by mostly getter systems, sometimes including spatter ion pumps or turbomolecular pumps. Okay, so what happens is that we measure this peak for the different gas species. We integrate the signal, we multiply by the sensitivity of the gas analyzer, and we multiply by the pumping speed of this conductance, which has to be lower with respect of the pumping speed that we apply behind it. Okay? And with these tricks, we can measure of the gassing rate. This is the most recent case. It is the comparison between as received, so not vacuum fired, austenitic and ferritic stainless steel. Is a work that we do for the gravitational wave beam pipes, laser beam pipes. So you see, for hydrogen, typical value of gassing rate of 3 or 4 is in the 10 to the minus 12 millibar liter per second. By the way, here we see also increasing the breakout temperature, that the degassing rate increase. This is very well-known phenomena. Is due to the hindering of the release by iron oxide on the surface of 304.
Sorry, it's a question. This graph intends the temperatures at 150 degrees.
Now, this is the bakeout treatment. No, this is room temperature. Room temperature.
Room temperature after bakeout of 150 degrees.
Yes.
Okay. Okay.
So while for the 404, which is an alloy of ferritic steel, where we have some niobium, titanium, some stabilizer of the ferritic structure. You see that the degassing rate that we measure are in the order of 10 to minus 15. And unfortunately, with the system that I showed before, we have an intrinsic limitation at, I think, 8, 10 to the minus 16 millibar liter per second. And what we measure is exactly the background. So this is the reason why we see that we put here the detection limit. Okay. Of course, all this has to be done measuring the background before, and then subtracting the value of the gas. The same has been done with the CH4. And with the CH4, the background level is much lower. And you see that we can measure value of something like 10 to the minus 17 millibar liter per second. So the order of few hundreds molecule of methane released per square centimeter per second. Okay, this is the way we do in general.
Yeah. Okay. Thank you. Thank you very much, Paolo.
I have a question, if I may.
Please.
Please.
How did you calibrate the RGA?
So the gauge is calibrated in an office that we
RGA, the residual gas analyzer.
Yes, sure. Residual gas analyzer is calibrated with respect to a Balmer gauge, which is calibrated by the PTB in Berlin.
Okay.
So
it's a transfer of
with pure gases.
With pure gases, yes. Let's say that we are operational oriented, not meteorological oriented. So for us, an error of factor of 2 is simply irrelevant. Okay. We are not meteorological. So for us to measure a degassing rate of 2 10 to the minus 15 or 3 10 to the minus 15 is nearly equivalent. What is important is that we can compare material with the same system.
Okay.
Thank you.
If there are other questions, we can do at the end of the all speakers. The next speaker is unfortunately I can't see still Yoshida from Japan. I'm sorry. The next speaker anyway is John Jansen, that is a research director at Institute on Membrane Technology in Italy, LCNR in Calabria. And we move in a topics slightly different, but anyway, because we are in the topics of membranes technology, but this is the scope, the aim of the workshop. So mixing different technology and different applications. John, this is the questions. Among your several notable works on membranes for gas separation and CO2 capture, I noticed those in which you used residual gas analysis in a vacuum, employing or accumulation method or throughput method to measure the permeability and diffusivity of polymeric membranes. Could you please share a few words to describe the advantage and disadvantages of these techniques compared to the other methods that not employed vacuum?
Yes, sure. Thank you first for the introduction. I feel a bit strange in this company because I see people speaking about 10 to the minus 14, 15, 16.
Yes. Yeah.
And we've been working for years on trying to get as high as possible permeability of polymeric membranes. So we are actually at the other side. I'm not sure I have one slide that I can show, but since I have two screens, I never know if I will succeed. Let me see. Indeed, I don't see it. Yeah, okay, yeah, maybe it's here. Okay. Share.
Yeah.
Yeah, you do see a screen. Okay.
Well, yes.
Yeah, what is the advantage of the method that we use? Let me first start with the traditional methods that we use. That's not this one. In this figure, I showed the one that we use a residual gas analyzer. But traditionally, around this membrane cell that you see in the middle is a vacuum system where you put vacuum on both sides of the membrane. Then at a certain moment, you expose the membrane to a gas, a single gas, which is one of the limits to the traditional system. Then at the other side, you're going to look, as soon as you expose the membrane to the gas, what's happening with the pressure. And from that, at a certain point, you can measure permeability, diffusion coefficient, and indirectly the solubility. I'm not going to detail, but that method is quite limited because you can only do it with single gases. That's so you can characterize materials, but just with single gases, and you will never know how a membrane will really perform if you're exposing it to a mixture. Well, at that point, we decided, no, we need something better. We still need to look at the transport through the membrane, but we want to see that with gas mixtures, and that means you need to make two changes. One is you need to take a setup like this one, where from one side you can feed a gas mixture, and from the other side, with or without a sweeping gas, you get a mixture through the membrane. And then you have to analyze the composition. The traditional method is using a GC, gas chromatography. Well, the faster micro GC are still slow. You need some tens of seconds to get a single measurement point.
Yeah.
Well, we would like really to see what's happening inside the membrane at every moment as soon as you expose it to the gas, or what happens if you change the temperature, if you expose the membrane from a dry gas to wet gas. You really want to see the reaction of the gas. And that's why we needed a faster gas analyzer. And for this purpose, we use a residual gas analyzer, basically a quadruple mass spectrometer.
Yeah.
We are not doing any GC/before because many people use a GCMS, but we send the mixture directly into the GC. So we have basically instantaneous measurement of the mixture of our gases. In principle, but of course not. Engineers among you will know when you feed I think, I'm not sure if it's showing this. Yeah, okay.
Yeah.
If you want to know what the membrane does, first you must realize that when you want to expose the membrane to the gas, the feeding system has some delay, and that's simply the flow rate, the volume divided by the flow rate of the piping. Of course, we optimize that. At the permeate side, we have the same. The volume of the piping divided by the flow rate. And then the slowest part is actually what happens inside the analyzer, because there's a capillary in the mass spectrometer to sample really small amounts. And that we found a way to quantify all that. We know the delay that's caused by the system, and then we can subtract the delay from all our measurements and finally come back to this time lag of our membrane, which then gives the diffusion coefficient, and that makes this system unique because everybody can measure a permeability. That's nothing really complicated. But if you are really able to measure the time dependence of your signal, then you can also get the diffusion coefficient of your gas through the membrane. And that's still not unique, but if you have a mixture and you measure with a mass spectrometer, you see both gases. You know what you feed, you see what you get out of it, and you can see the time dependence, the transient phenomena of both gases in the system, and from that you can calculate the diffusion coefficient of two gases that move at the same time through a material. And you understand how they influence each other, because very often they influence each other. That can be positive or negative. Usually a gas like CO2 drags the other gases through the membrane, so the diffusion tends to get faster. But CO2, if you have high free volume polymers, CO2 occupies a lot of volume. So you might reduce the solubility of the other gases. And the balance between that, you never know what's going to happen. And that's why we study a lot of different materials. You never know what's going to happen. But in some cases, the selectivity of we study CO2 over other gases because we look at biogas separation, biogas upgrading, flue gas separation, oxygen, nitrogen separation. You never really know what's going to happen in a real system. And that's why it's very important that we can actually see it with this system. That's basically in a nutshell what we are doing. We study this on a lot of different materials, from perfluorinated polymers years ago already, recently ionic liquids, very interesting materials from several points of view. And in the membrane world, since some 20 years, the polymers of intrinsic microporosity are under observation. They are very interesting because of their incredibly high permeabilities. Also a lot of other difficulties, but these are very interesting materials for studying. And that's what I'm going to speak about in October.
Yeah, thank you. Very, very interesting, yes, because yes, you can measure also the solubility with this setup, because you measure diffusivity, and it's mandatory to use a residual gas analyzer because gas chromatography, if I understood correctly, it's not enough fast, no?
No, even the faster, they need 10 seconds or so for every single point. While if you have a good system and you optimize it and you have a sensitive mass spectrometer, it can take points per second. So we are a little bit slower, but still we can measure every few seconds a point. And that means that transient phenomena that take a minute or so, which are not uncommon, we see that over the whole curvature of the signal. So it's a really powerful method.
Very interesting, John. I hope to discuss again of these setups and of your need in October together.
Yes.
Anyway, for questions, I cordially ask to wait the end of the speak of Orlando, that it is invited guest after John. Anyway, maybe Hajime Yoshida, in my opinion, could have a wrong with the hour. But anyway, maybe will be appear at 3 o'clock. I don't know. Anyway, joining us now with Orlando Teodoro. Orlando Teodoro is a proof at the center, professor at the Center for Physics and Technology Research at Physics Department, the Faculty of Science and Technology in the University of Lisboa, in Caparica, exactly. Orlando, can you see me?
Yes.
Okay. Orlando will speak with us about cork. Very interesting, another very interesting topics. You have expertise in the metrology and modeling of gas transport through cork closure. I'm curious to know what are the main mechanisms of the gas transport in these materials, Knudsen flows or phase diffusion or solution diffusion. Okay.
Okay. Thank you very much for this opportunity and for your question. Yes, some time ago we did some work on cork, and more recently on wall cork stoppers. And one of the things we addressed was how the gas how the gas were transported in cork. We used small slices to measure the permeation, and to answer quickly to your question is that we found Knudsen diffusion mechanism for transport. When we change the gas type and we plotted the permeation as function of the inverse of the square root, we found a linear dependence. And so we are convinced that for dry, let's say dry gases, this is the model. However, for vapors as ethanol and water, they are very far from this line. And we believe that we have a second mechanism by solution diffusion. Water, for instance, has a huge affinity with the cellulose, which is one of the main components of cork. And so it is understandable that we have another route for water transport. But for dry gases, and oxygen is one of is the most interesting for this application. We believe at least our data is consistent with this model. So cork, just to remind, is like a foam. It's a series of empty cells with gas inside. They are they are tight, they are flexible, but they are closed. They are not, what we see in this picture is not pores. We cannot call these pores. They are cells, they are closed. But they connect each other by small channels in nature, in plant cells. These channels between cells are well known. They are called plasmodesma. And so if some of these channels, small channels below 100 nanometers are open, are unclogged, the cells of cork are empty of cytoplasma, so the cell is the inner part of the cell was removed during cork growth, and only air remained inside. And so if some of these small channels are still open, so they can explain this nice dependence, this Knudsen diffusion as we observed. But besides measuring the permeability, there are at least 3 key challenges that are very, very important. Many other authors measure the permeation, the permeability of cork, and they also found a huge sample-to-sample variability. So we can find samples, even selected carefully, the samples. So here I'm talking about slices, carefully selected without any visual defect. But even so, we could find distributions spanning over 2 to 3 orders of magnitude. That was very difficult to take conclusions on standard deviations, since in these cases the standard deviation was even higher than the mean, and so not much conclusions we can take from that. However, in wall cork stoppers, the scenario, the data is better, and since the cells are compressed and they span over less orders of magnitude. But in that case, we believe that the measurements that we can make in wall cork stoppers are due to leaks, not to true permeation. The interface between glass and cork is very tricky, is very rough. Cork is rough, although flexible, and it is the flexibility that gives the tightness, the sealing capacity. For liquids is easy, but for gases is more difficult, and some routes can remain open through. So we see here a section of a bottle. So this green part is the top of a bottleneck, still with the cork inside, and then we took this sample to a SAM, and these images are from the SAM, and we can see the glass and the cork and the interface, and there's some parts that are not smooth, but we can see that the interface is not continuous. And so understanding what is leakage through the interface and what is true permeation, it is tricky, and we believe that many authors were not able to separate these two phenomena. And another thing that is a challenge is to to do this in a traceable way and not necessarily with oxygen, but somehow with a gas that can be converted to oxygen. That's what we did. And in as fast as possible. Some of these measurements may take months to get. And so we believe we found, we refine a technique to do it in a few hours or a few days eventually. But we could be sensitive and reproducible with cork. So just to situate us in the measurements, so we saw from Paolo numbers in 10 to minus 14 millibar liter per second per square centimeter, if I remember.
Oh, yes.
Here it is below 410 to minus 8 millibar liter per second. And that this is the range, at least one order of magnitude below.
Yeah.
Okay, that's it. Thank you.
Very interesting. It's also in particular for the Italian wine company. This is this study. Yeah. Unfortunately, we can't see still our Japanese colleague. Now it's time to your questions, and I can see the first question in a chat by Julian Gargiulo. Julian, if you say your questions, or you prefer that I speak, is a question for Paolo and Carlo. Julian asked, thanks for the presentation. Okay, the microphone doesn't work. And ask why we have chosen methane, because it's considered predominant as outgassing besides hydrogen. Paolo or Carlo?
Maybe can just simply answer. In fact, I showed two. Show the hydrogen and methane. Okay,
Yeah.
which I mean, hydrogen is very important because it's a dominant gas released from steels, whatever steel, and methane because methane is not pumped by non-evaporable getter, and today accelerators and in the future probably gravitational wave systems will be massively pumped by non-evaporable getter. So the fact that I show CH4 is important because it allows to design the pumping speed, which is not a non-evaporable getter. For example, ion pumps. Okay, so these are the most important gases. There is also similar graphs for CO and CO2. Even if I have to say that the precision of the measurement is questionable, because for these two gases there is a slight but significant amount of gas which is condensed on the wall of the system. So we lose a part of the gas which is accumulated. Okay, and so the measurement is certainly less precise. But okay, for the accelerators or gravitational wave beam pipe, the two most important gas are hydrogen and CH4.
Okay, yes, exactly. Me too, I have questions for you, Paolo, if you can answer. Regarding the problem of hydrogen, energy hydrogen, I know that for hydrogen storage there is a problem of stainless steel embrittlement for hydrogen. You have ever addressed this problem, or or not? Embrittlement like for the neck, no? The same effect also for hydrogen, also for stainless steel.
In the application that in which we work, there's not a lot of hydrogen.
Yeah, yeah.
I mean, it's not. I know
Yes.
that I have colleagues working for ITER, and there, in fact, there is a this problem of embrittlement to be considered. I'm not an expert, but I can say that ferritic steels are in general more sensitive to embrittlement than austenitic.
Okay. Okay.
What, in addition, I mean, what is important for us is not really embrittlement, is trapping.
Okay.
Because in the ferritic steels, the diffusivity of hydrogen in the lattice is so high that if you have inclusions, if you have precipitates, you have always hydrogen which is trapped around. When you do thermal desorption or thermal extraction spectroscopy, what you see are tiny peaks, okay, increasing at speed which is not enormously high. I mean, I'm saying something about 5 degrees per minute.
Yeah.
You see very tiny peaks that do not scale with the thickness. So they are invariant with respect to the thickness, which means that is typically not a diffusion related, but is detrapping related.
Yeah,
This is
surface. Yeah, sure. Okay. Well, other questions? If you don't.
Maybe I've got a question again for Paolo. I was actually typing it, but since you ask, I can directly ask the question. So you showed that you transfer ion current into pressure or mass flow by using the sensitivity of the RGA. Is that sensitivity that you pre-calibrated, or do you determine the sensitivity directly with the pressure gauge? Or
no. We have to measure, of course, the sensitivity of the RGA with respect to typical peaks related to a certain gas. For example, for CH4, normally we use 15, which is typical, okay? And then we can use some analysis to deconvolute the different peaks. But okay, this is the way. So what is important is that to realize that the ratio of the sensitivity for a specific RGA in general remain constant. It doesn't matter how the absolute value of the sensitivity evolves. So what do we do in general to verify that we don't have a drift in sensitivity? We inject a gas just before the measurement. In general, is argon. Okay, for example, if the system is massively pumped with a non-evaporable getter, argon is a choice because it's not pumped by the getter. Okay, and with that we compare the reading of the ion gauge with the one of the mass 40 of the RGA. Okay, so if the sensitivity changed by 15%, we automatically change the sensitivity of all the other gases with respect to the ratio of the 1 of the 40. Okay, so this is just to monitor that you don't have drift. I mean, I think if you use RGA, you know that this is a typical problem of RGA is that they have a drift. In particular, mainly if you use electron multipliers. It depends on the environment, how many hydrogen you have, oxygen, etc. So yes, if you want to have a good measurement, you need to integrate in the system an injection of a certain gas. Another way to bypass the cross-check with the ion gauge is to have calibrated leaks. Okay, so you inject a calibrated leak. Okay, then you know the configuration of the system, and if you want to be very precise, students like that, you can do a full Monte Carlo simulation with mole flow, exactly know what is the signal of this calibrated leak at the level of the RGA. and then you adapt. And
yeah, okay, thanks. Yeah, that's I was wondering indeed what you do to determine the drift, but that's clear now. Yeah, thanks.
Okay. We are lucky because it's appeared Hajime Yoshida. It was, I can imagine, in my opinion, for in Japan, 2 or 3 o'clock. Hajime, you are with us? Hello, Hajime. Sorry for I mispronounced your name. Can you? Your microphone works?
Can you hear me?
Yes, now yes. Now yes. Hajime, we have the last question is for you because we have started 2 o'clock in time of Rome. No? For you at 10 o'clock on 11 o'clock post meridian now in
So
Japan.
you mean maybe I'm late this webinar?
Yeah.
I am sorry.
No
maybe
problem. No problem. There is time for your questions and for you.
Yeah.
Have you the possibility to display your slide, or otherwise I do it?
You can introduce. You
Okay.
already introduced my presentation, right?
Okay. Now I introduce you to other people.
Okay.
Hajime Yoshida is the head of the Pressure and Vacuum Standard Group of the Research Institute for Engineering Measurements and National Metrology Institute of Japan.
Yeah.
My questions to Hajime is this one, related to the water vapor. One of the most important characterizations for film used in food packaging, but not only, a lot of other applications, is the water vapor transmission rate. A high vacuum experimental setup equipped with a residual gas analyzer can be employed for this purpose. However, water vapor tends to have a high absorption rate of the inner wall of the chambers. When testing very low permeability value, what is the best method to minimize or account for this drawback? Okay. If you want, I can share the, okay, I can share for you. Can you see the slide of the Hajime?
Yes, I can.
Okay. This is the questions, okay? So I move to the next slide. Please.
Yes. You know, there are 2 types of WVTR, water vapor transmission rate measurement methods using vacuum technology. One is accumulation method and the other is throughput method. What they are in common is water vapor is admitted in here, and film is located between the water vapor and vacuum condition, and water vapor permeates through the film is measured by detector. The difference is the following. In accumulation method, the valve between vacuum chamber and vacuum pump is closed. Then the pressure in the vacuum chamber is keep increasing, which causes integral type permeation curve. The WVTR is calculated from this slope. In throughput method, the vacuum chamber is continuously evacuated by vacuum pump. Then the pressure in the vacuum chamber first increase and finally reach the stable saturation. WVTR is calculated from. Please go to next slide. Adsorption is different between 2 methods. In case of throughput methods, left hand side, the in the pressure equilibrium is realized, which means the desorption rate onto the vacuum chamber is balanced with the desorption rate from the vacuum chamber, which means the net adsorption rate becomes zero. In other words, influence of water vapor adsorption is cancelled out due to realizing equilibrium pressure. Therefore, accurate WVTR measurements can be achieved. In accumulation method, however, the water vapor pressure is keep increasing. Therefore, the adsorption rate onto the vacuum chamber is always higher than the desorption rate from the vacuum chamber. Therefore, the net adsorption of water vapor may cause the underestimation of the WVTR. Please go to next slide. This graph shows the comparison of 4 types of WVTR measurement methods. QMS, right-hand side, QMS shows the result of throughput method. PS1 and 2 shows the result of accumulation method. GC2 means a gas chromatography method. It's a kind of accumulation method. IR1 and 2 shows the result of MOCON permatran device, which is a kind of de facto standard. 3 reference films with a small hole were used as samples. Red points show the result when 10 mm in diameter small hole with the reference film with 10 mm small hole were used. Similarly, blue and green points show the result when 3 mm in diameter and 1 mm in diameter small hole were used. And 3 dot lines show the calculated WVTR from the inner diameter of pinhole, a small hole. As shown here, the result of throughput method is close to the calculation values showing dot lines. While the result of accumulation method tended to lower than the calculated value. I think this result shows the throughput method is more reliable than accumulation method in case of water vapor transmission rate measurement. Please go to next slide.
Yeah.
However, throughput method still have issues related to water vapor adsorption. First is QMS signal does not become perfectly stable at the region. Therefore, it is unclear when WVTR has truly reached saturation. Yes, and the signal is typically drift. Second, it is difficult to distinguish between 2 processes, A, diffusion of H2O molecules through the films, and B, adsorption of water vapor on the chamber wall. And this makes challenging accurate evaluation of diffusion coefficient. The method to minimize or account for these drawbacks is a question I've received from Giuseppe.
Yeah.
Yes, it is very difficult question. I don't have a clear answer. But in general, selection of material, temperature, surface treatment, piping, and the structure of WVTR measurement device is important. Also, using well-characterized reference films are also useful. But I don't have a clear answer yet. I'd like to find some tips at this workshop. Thank you.
Have you ever heating the chamber to avoid to decrease the absorption rate of water during measurements or not?
Yes, I'm typically keeping the temperature of vacuum chamber at 40 degrees C.
Okay.
40, a little bit higher than the room temperature.
Mm-hmm.
I try to I have tried to temperature higher, but it doesn't work well. I don't know.
Yeah. Yeah, I know.
Mm.
It's a challenge, very challenging. Yes, it's very challenging. Yeah. Anyway, thank you, Hajime.
Yeah.
Have you okay? Have you questions for Hajime or for other invited guests?
Yeah, maybe I have a question.
Yeah.
Because we have the same problems also for measuring the gassing rate of water when it is low. So the question is, have you ever thought about or tried coatings that can reduce the sticking or the absorption of water? So we tested with a limited success silicone coatings deposited by CVD.
Good idea.
And in fact, we can reduce by a factor of 10 the degassing rate of water, which is good, but not enough to avoid the bakeout for us. Okay. Is there any chance that there is a coating that can help you?
Thank you for your question. I've investigated many research papers about these techniques. But I have not tried yet. I am just using stainless steel, but it is polished like a mirror. Yeah. It's all from my experience. But I have heard silica coatings sounds good. Yeah.
Also, in my opinion.
Thank you so much.
Also, in my opinion. Good idea, Paolo. Good idea. Yeah. Other questions? Okay. Let's say yeah, we have complete 1 hour. I wish to thanks again all our invited guests. And just a reminder that you'll have the chance to meet each of them in Portofino in this October and dive into the topics you're interested. In my opinion, these topics has different approach, but the vacuum community can exchange expertise with membrane community in order to solve problems. Could be an advantage for both community to share its open questions, we say.
Yeah.
And this is for the reason of organizing this workshop. In my experience, I found difficult to speak with membranologists instead of vacuum scientists. Instead, to speak the same languages could be an advantage for both community. So, if you don't have other questions, I thank you all again and see you in Portofino in next October. Thank you. Thank you
Thank
all.
you very much.
Thank you.
Bye.
Thank you.